Welcome back to Biomandscape. Today we are going to know the process of gene cloning. So gene cloning is a process of producing many identical copies of a specific gene by using a cloning vector like plasmid. It happens commonly in E. coli inside a host cell. So first what happened? The isolation of the gene of interest. So what happened? First we extract the DNA from the donor organism, then identify and isolate the desired gene using a restriction enzyme or a PCR. Then a plasmid vector is isolated from the bacteria. The vector contains an origin of replication and there is a selectable marker like antibiotic resistant gene, and multiple cloning sites are present. First, the cutting with the restriction enzyme takes place where the gene are and the plasmid they are cut with the same restriction endonuclease, and this produce a compatible sticky ends or blunt ends. Then the gene is joined to the plasmid using a DNA ligase, and this form a recombinant DNA molecule. Then the recombinant plasmid is introduced in the competent bacterial cell using the E. coli, and this method include the heat shock treatment or electroporation. Then the cell are grown on an antibiotic containing medium, and only bacteria carry the plasmid survival. The recombinant these colonies are present recombinant colonies. They are identified by technique like the blue white screening or colony or PCR, and the recombinant bacteria they divide rapidly, and each cell copies the recombinant plasmid and it produces multiple millions of copies of a desirable gene. The clone gene can be expressed to produce a protein like insulin or isolated in a large quantity for sequencing, research or genetic engineering. So first, what are the steps? First, gene of interest plus the plasmid vector is taken. Then restriction enzyme digestion takes place where the restriction enzyme cuts the DNA. Then DNA ligase, it joins the DNA fragment. Then recombinant plasmid then transfer into the E. coli. What does the vector do? It carries the foreign gene. Select a recombinant colony, then bacteria will multiply and millions of gene copies are obtained, and or protein is produced. So what is the role of the vector? To carry the foreign gene. What is the role of the host cell? To multiply the recombinant DNA. At the ORI site, the plasmid replication is begin, and the selectable marker it identify the transform cell, and blue white screening it distinguish the recombinant from the non-recombinant colony. So for knowing such concepts, follow Biomandscape.